part 2, we're going to continue with our, what are they called? The special products derived from the amino acids. Previous video we talked about heme, now we're going to talk about creatine. So creatine is present in muscle, brain, and blood, both as phosphocreatine, also known as creatine phosphate, and in its free form. So trace amounts of creatine are normally found in the urine. Creatine phosphate is a reservoir of high-energy phosphate bonded to it. When ATP concentration is depleted or reduced, the creatine phosphate generates ATP from its pool within the skeletal muscle during contraction. The phosphate group transfer potential of creatine phosphate is higher than ATP, essentially meaning that you can get more bang for your buck with creatine phosphate than just with regular ATP. When talking about creatine synthesis actually begins in the kidney and is going to be completed in the liver. And there are 3 amino acids that we're going to look at. We have glycine, arginine, and then methionine. We look at our SAM, that S-adenosyl homocysteine. Remember our SAM we talked about in the previous chapter. I believe it was chapter 8 that we talked about SAM. Let me double check which chapter that was. Yeah, it should be chapter 8, that 1 carbon metabolism. So that's going to come back here in a second. So within the kidneys, so glycine is going to combine with arginine to form I love pronouncing these, guanidoacetate, and ornithine, which travels to the liver. So we produce some ornithine and the guanidoacetate right there. In the liver, guanidoacetate is methylated by our S-adenosyl methionine, also known as SAM, in order to form creatine. And then it is released in the bloodstream and enters into the cells. So there we have our combination of glycine, arginine, and the methylation is our methionine in order to create creatine. Creatine is phosphorylated by taking the terminal phosphate group of ATP to produce creatine phosphate, which is an unstable compound and can undergo what's called spontaneous cyclization, forming creatinine. So little devil's in the details with this. Creatine, right there, we have at the beginning part, so we bring all this stuff to create creatine. And creatine travels off to the brain, the heart, and the skeletal muscle, okay? Now, inside of the cells, we take the terminal phosphate group of ATP. So we take the phosphate off of ATP to produce creatine phosphate, which can undergo spontaneous cyclization, forming creatinine. So slightly different language here, which cannot be further metabolized and therefore is excreted in the urine. The reaction is catalyzed by creatine phosphokinase, also known as creatine kinase. So there's going to be our enzyme. So creatinine, if you see creatinine, this is going to be one of our end products. Kind of our leftovers after we use the creatine, okay? This is also you'll learn this later. There's a test called creatinine clearance test, and that's going to be a test that tests our kidney function. You'll learn more about that when you get into the actual testing, like the lab testing and stuff courses, and we start talking about kidneys and kidney disease and all kind of stuff. So Phys 2 as well as in Tri 4 and Tri 5, you'll get into some of that. So between creatine and creatine phosphate, this reaction is reversible, and And the skeletal muscle generates ATP from creatine phosphate during muscle contraction. Creatinine is what's called the anhydride form of creatine. It is formed largely in muscle by irreversible, keyword there, irreversible, non-enzymatic degradation of creatine phosphate. A trace amount of creatinine is excreted in the urine. So we have our creatine phosphate. So it can go back and forth between creatine and creatinine phosphate. Essentially, the creatinine phosphate is holding on to a terminal group of ATP, and it's what's going to give that up to an ADP to form more ATP, and then it goes into creatine, and it can go back and forth as it continues, to kind of reset, recharge. Once it's all done, once it's used up, you can get the spontaneous cyclization. So essentially the breakdown of creatine phosphate, and it turns into creatinine, which is that end waste product. So creatinine excreted per day depends on body muscle mass and kidneys function right there. So in case of kidney failure, creatinine levels increases in the urine. Now, next part, synthesis of purines and pyrimidines. The dietary uptake of purine and pyrimidines bases are low. They are synthesized de novo, which just means they're synthesized from scratch, they're synthesized anew, or they are salvaged from existing bases. So the synthesis of the purines and pyrimidines occurs in the liver, as well as the brain. Liver is often the number one, brain is number two of the synthesis. So then the bases are transported to other cells for nucleic acid biosynthesis. The synthesis of the purines have a whole bunch of different parts. Glycine, ribose 5-phosphate. Remember ribose 5-phosphate from our pentose phosphate pathway. We had broken all the way down. We got the NADPHs, we got the xylulose 5-phosphate, and then the xylulose 5-phosphate gets broken down or gets converted into ribose 5-phosphate. So we got all those. We need glutamine, we need aspartate, we need carbon dioxide, and we need N10 formyl tetrahydrofolate. Tetrahydrofolate, right there. If we remember, what is that coming from? Tetrahydrofolate. Folate is going to be our B9 vitamin. It's coming from a component of it's coming from our B9. So our ribose 5-phosphate, we get ATP added into it, and we'll just go ahead and read down here exactly what it is. So 5, again, big long words, we'll just shorten it to PRPP. 5-phosphoribosyl 1-pyrophosphate, PRPP. When you're doing these, kind of writing these pathways out on that cheat sheet that I told you to do, write it out once and then use the abbreviation. You want to be able to recognize the full name if it's asked, but most often in questions and stuff for these really long stuff, he will occasionally use the abbreviated form. Okay, so the PRPP is the source of the ribose moiety synthesized from ATP and ribose 5-phosphate from that pentose phosphate pathway. The purine bases are built on the ribose sugar. The purine nucleotide biosynthetic produces inosine 5-phosphate IMP in 11 different steps, which we'll see a lot of these steps. From what I recall, he doesn't get super specific with all of these steps, from what I recall. I don't know if he's changed his testing strategy or if he goes more into this. But again, I wouldn't focus too much on memorizing all of this stuff. I'd focus on the big components, focusing on the precursors, kind of what amino acids there are, where you're getting these different components, and potentially kind of in what order you're using them is what I would focus on. Okay, if you can learn the whole thing, even better, but again, just what I would focus on. So the ribose is activated in a reaction with ATP, forming the PRPP. PRPP then reacts with glutamine, which forms phosphoribosilamine, which is catalyzed by an enzyme called PRPP, glutamyl aminotransferase, also called glutamine phosphoribosyl aminotransferase. The amide nitrogen of glutamines, remember glutamine right there is an amino acid, so our amino acids the carbon has that nitrogen on one side, the other carbon, the R group, and the hydrogen, so that amide group becomes N9, so it's the ninth, it's the nitrogen in the 9 slot, in the 9th position. This enzyme is the rate-limiting enzyme in purine biosynthesis. Right there, ding, 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 ding. Pay attention right there, rate-limiting enzyme. We need to remember this name, put a big star by it, because that's more than likely where our big question for this chunk is going to come from. And it catalyzes the formation of phosphoribosilamine. So would I know the rate-limiting enzyme, know what's right before it, know what comes right after it, know what it uses, what goes into it, this being the glutamine. So those are the kind of, specifically, those are going to be like the main questions he's going to ask with rate-limiting enzyme stuff. A molecule of glycine is then added, which provides C4, C5, and N7, and forms glycinamide ribotide, also known as GAR. Then the N10 formyl tetrahydrofolate donates 1 carbon to GAR and contributes for C8. We just keeps on going. The amide nitrogen of a second glutamine is added, which contributes for N3. C6 is added in a carboxylation reaction. Aspartate contributes N1 by forming an amide bond with C6. N10 formyl THF adds one last carbon moiety, which becomes the C2 in the purine ring. These reactions finally produce the first purine nucleotide, IMP, in this biosynthetic pathway. IMP is rapidly transformed to AMP and GMP, those nucleoside monophosphates. So again, there's a lot of steps kind of in all of this, a lot of big names. What I would focus on is know kind of what's going in where and kind of the rough order of stuff. You don't necessarily need to remember each of these individual steps, and a lot of this, like the N3, the C6, the N1, all that kind of stuff, it's a pretty overkill to remember all of that. Remember the enzymes, remember the rough order that things go in, and kind of what you get out of it is what I would focus on, okay? There's our IMP. This is just showing this reaction, how we're going to go to our AMP or our GMP, or if we think about we remember when we talk about DNA, RNA, we have our G, our A, our C, our T. Remember, G goes with C, A goes with T. So right now with this, we just talked about the A and we talked about the G and the formation of those guys. Again, here are some more of those enzymes. Using aspartate, using fumarate, and how they're converted, how the NMPs are converted to di and triphosphate derivatives in 2 steps. by nucleoside monophosphate kinase and nucleoside diphosphate kinase, respectively. I don't know exactly where this fits in to this, but if it comes back, we'll look at it. Okay. So the brief outline for purine biosynthesis: ribonucleotide reductase, an iron-containing enzyme protein, is required for the synthesis of the deoxy form of nucleotide diphosphates, deoxy ADP, deoxy GDP, in purine biosynthesis. Note here, the substrate for the ribonucleic reductase are the nucleotide diphosphates ADP and GDP in purine biosynthesis. So these are the substrates. So essentially these formation of the AMP and the GMP, the substrates, because we know they need to be further processed in order to be involved in DNA itself, because it's a deoxyribose. So this is just adding that extra little step that we have a ribonucleotide reductase is required in order to get them to that next step. I would remember this enzyme, just that it does the deoxy part, and that should be good enough for the test. Now, the pyrimidines. Pyrimidine biosynthesis utilizes 3 precursors. These are aspartate, which contributes to the N1, and again, all of these numbers afterwards are the position. So it gives a nitrogen that happens to be in the first position, a carbon in the 4th position, a carbon in the 5th position, a carbon in the 6th position. 1, 2, 3, 4, 5, 6 positions in this specific area of this pyrimidine ring. Okay? Bicarbonate contributes for C2, and then a glutamine donates N3. So what you'll want to remember aspartate, remember bicarbonate, remember glutamine come together to form the pyrimidine. In the pyrimidine nucleotides, the base synthesized first, and then it is attached to the ribose 5-phosphate moiety. Remember that ribose 5-phosphate is coming from the pentose phosphate pathway. In the first reaction, glutamine reacts with CO2 and ATP to form carbamoyl phosphate, catalyzed by another big ol' enzyme, cytosolic carbamoyl phosphate synthase 2. This is an enzyme that we have seen before. Our systolic carbamoyl phosphate, and kind of in that carbamoyl phosphate process. It was seen in chapter, I want to say it was chapter 7, I believe we talk about that. We talk about the whole process about in the mitochondria to the cytosol. Okay. This enzyme is the rate-limiting enzyme, so right there we want to make sure we remember this enzyme in the pyrimidine biosynthesis pathway, and is inhibited by UTP and activated by PRPP. Inhibited by UTP and activated by PRPP. In the second step, an entire aspartate molecule is added to the carbamoyl phosphate. The resultant product is then closes to produce a ring, which is then oxidized to form orotic acid, also known as orotate. Anytime you see tate at the end of something like that, you can know it's also an acid, like glutamate, glutamic acid, stuff like that. Orotate reacts with the PRPP to produce orotidine 5-phosphate, OMP, which is then decarboxylated to form the first pyrimidine nucleotide, uridine monophosphate. So another thing that we'll want to remember is what the quote-unquote, like the original of each of these is. The original purine was the IMP. Make sure where to say exactly what that's called. The inosine 5-phosphate is the original purine. And then the original pyrimidine is the uridine monophosphate. Okay? Then UMP is converted to UTP by nucleoside monophosphate kinase and nucleoside diphosphate kinase successively. And then CTP is synthesized through amination of UTP by glutamine. So this is like the whole process. Again, if you can remember the whole thing, great. If not, make sure you remember what is going into it. Okay, ribonucleotide reductase. Again, we saw that one before. This enzyme catalyzes the conversion of the ribosugar moiety to the deoxyribose. So converting the CDP to DCDP. So again, this is also iron, because we saw that earlier that ribonucleotide reductase is an iron-requiring enzyme. If we're low on iron, it can also decrease our nucleotide biosynthesis. Now the degradation pathway, breaking these down, breaking the purines down. Breakdown products of purine and pyrimidine bases synthesizes uric acid and urea respectively, which we already talked all about the uric acid and urea cycle. Let's go ahead and run through these. So the 5-nucleotidase catalyzes the removal of phosphate group from GMP and AMP, resulting guanosine and adenosine respectively. Guanosine is further converted to form guanine by releasing the ribose sugar, followed by deamination to produce xanthine. Adenosine is converted to inosine by deamination, then releases ribose sugar to form hypoxanthine, followed by its oxidation to form xanthine by xanthine oxidase. You can follow this whole process here, AMP to adenosine, inosine, hypoxanthine, and they both go to form xanthine, and then go to uric acid. Again, I don't quite remember how much he gets into the specifics of all of this, of the degradation part. Do your best to remember as much of it as you can. Again, I'd probably remember the big ones, that they both go, that they both go to xanthine, they both go to the uric acid, to urea. That's the stuff that I would focus on recalling. If you can remember the whole thing, great. If not, pick the key components of it to hopefully remember that. And then so it forms the uric acid, and the uric acid is an excreted end product of purine catalysis in mammals, including humans. Pyrimidine catabolism leads to the formation of ammonium ion and eventually to the production of urea. Thymine degradation product is methyl malonyl semaldehyde, which is also an intermediate in valine metabolism. There's kind of a bunch of extra stuff in here. And that product is catabolized to form succinyl-CoA, beta-alanine, and beta-amino isobutyric acid are the metabolic end products of cytosine, uracil, and thymine. I feel like if it does come up, like this might have one question, this will probably have one question on it. Is it really worth it to memorize this entire process just for those one questions? Probably not. I'd probably remember pyrimidine degradation, connect it with methyl malonyl semaldehyde, and then connect it with succinyl-CoA. That's about as far as I would go into remembering that component. Okay. I'm actually going to go ahead because, let's see, how long is tyrosine? Tyrosine has a couple different, tyrosine and tryptophan. I think we should be able to get through at least one of these before the half-hour mark. Okay, so the tyrosine is amino acid precursor for the synthesis of the following compounds. Now, tyrosine, tryptophan, 1000%. These questions are on board exams. So please, please, please pay attention to these and put these into long-term memory. So tyrosine gets converted to a whole bunch of different things. Some of the neurotransmitters, including dopamine, norepinephrine, and epinephrine. And these biogenic amines described above, dopamine, norepinephrine, epinephrine, are known as catecholamines. So these guys are, just know, aka catecholamines. Sometimes they'll try and trick you with questions saying like, which of the following does tyrosine go into, and it'll have like dopamine, norepinephrine, catecholamines, all of the above. And if you don't remember that one word, it can mess with you. And we also do skin pigment with the melanin. And then, of course, the tyrosine is the precursor of our thyroid hormones. And as we know, this specific professor does like thyroid hormones, so more than likely that is going to pop up. So we can see right here, phenylalanine. Even taking a step back from tyrosine, even a step before that, we have phenylalanine. So that'll probably play a role in this as well. So we go from phenylalanine to tyrosine, okay? And then the tyrosine can go right off, and it gets converted to technically goes to dopamine, and then it goes to the melanin, turns into melanin. But anyway, so tyrosine goes to dopa goes to dopamine goes to norepinephrine, and norepinephrine can go then to epinephrine. I think it got cut off down here, but norepinephrine turns into epinephrine. And this is just telling the different locations of this. So the neurons, we use the dopa, we use dopamine, we use the norepinephrine. And then in the adrenal glands, we have all of these as well, especially the norepinephrine and the epinephrine, which our adrenal medulla is releasing both of those. I wouldn't get too fixated on like the specifics. I would focus on this component up here, know what stuff tyrosine turns into, and kind of the flow. So phenylalanine to L-tyrosine to dopa to dopamine to norepinephrine, and technically off of dopa goes to the melanin. Catecholamine biosynthesis are described schematically as described in the figure above. Tyrosine also produces melanin in melanocytes. So here, L-tyrosine, L-dopa. This goes into a lot more detail than I ever remember him talking about. I'm not going to spend a lot of time on this, because I don't remember him ever going this deep. If he did in the lecture, I would go back and look at the lecture. I'm not going to spend a lot of time going through this entire schematic. I would focus more attention on this guy up here compared to this down here. Also, our thyroid hormones, thyroxine or T4, triiodothyronine or T3 are derivatives of the amino acid tyrosine bound covalently to iodine as shown below. Two tyrosine molecules are bridged with oxygen linkage followed by iodination. So here we have our tyrosine, 3-monoiodotyrosine, 3,5-diiodotyrosine. So there's one iodine bound, two iodine bound. Triiodothyronine, so there's 3 iodine bound, and then 4 iodine bound, known as thyroxine. Things you'll want to remember is that the thyroid hormones, the base is tyrosine, and just how many iodines are attached to it. You may want to look and remember the mono and di, but really just remember it's mono, meaning 1, iodine, tyrosine, di being 2, iodine, tyrosine, so tyrosine with 2 iodine, essentially tyrosine. This is thyronine, but tyrosine with 3 iodine, and then tyrosine with 4 iodine. How much do we have left? we'll just go ahead and finish it on this. We'll go a little bit over again on this one, but that's okay. Now, tryptophan is the precursor of the neurotransmitter serotonin. Tryptophan also produces melatonin. Again, remember, melanin in the skin, melatonin for sleep, which is produced in the pineal gland. Another thing that often what professors will do is they'll give you the answer choice of the pineal gland or the pituitary. These are different, okay? The pituitary gland is more in the front. It's located essentially right in, within the cella turcica, the pituitary gland is. The pineal gland is deeper within the brain, different portion of the brain entirely. They'll put pituitary gland in there to confuse you or to throw you off. So just remember, it's the pineal gland, not the pituitary gland. So melatonin is the hormone produced in the pineal gland, which regulates the circadian rhythm, day-night cycles, or light-dark cycle. Tryptophan also generates the cofactors nicotinamide nucleotides, or NAD+, NADP+, those guys. So from tryptophan to 5-hydroxytryptophan to serotonin to N-acetylserotonin and then to melatonin. Again, you don't have to remember all of the super specifics of it. I'd remember each part. So tryptophan goes to hydroxytryptophan, goes to serotonin, goes to acetylserotonin, goes to melatonin. That's what I would remember going across. Glutamate serves as the precursor for the inhibitory neurotransmitter gamma-aminobutyric acid, aka GABA. The enzyme glutamic acid decarboxylates glutamate to produce GABA. This reaction requires pyridoxal phosphate as cofactor. So things we'll want to know: glutamate gets turned into GABA. So remember, in the spinal cord we talked about glutamate activates. That was more in physiology. We talked about that. So typically glutamate by itself is an excitatory neurotransmitter. When it gets decarboxylated, then it turns into GABA, which is an inhibitory one. So we'll want to remember the enzyme, the glutamic acid decarboxylase, and that we need a pyridoxal phosphate as a cofactor. Again, that is our B6. Glutamate, our PLP, acted on becomes GABA. Histidine is the precursor for histamine. Hopefully those sound similar enough that you can remember that. Histamine is going to be released from inflamed or injured cells. Inflamed? I don't even know if that's a real word. Sounds cool though. Histamine is responsible for bronchoconstriction and vasodilation. This is almost always a question because it trips a lot of people up, if not in biochem, in physiology, or in general pathology. They love this kind of question, that histamine is bronchoconstriction but vasodilation. It increases blood flow but makes it more difficult to breathe. The histidine is decarboxylated to form histamine, and the histidine carboxylase catalyzes this decarboxylation reaction. So again, remember the enzyme. Pyridoxal phosphate, again, is the cofactor, so I'll want to remember those guys. Most often, pyridoxal phosphate is the cofactor that we're going to be using. That's wrong. It should say coenzyme, not cofactor. Because cofactors are metals, it should be coenzyme. Pyridoxal phosphate is the coenzyme. Arginine produces nitric oxide, a chemical messenger molecule that regulates several signaling mechanisms in our various biochemical reactions, primarily vasodilation. The reaction is catalyzed by NO synthase, nitric oxide synthase. It is produced by the vascular endothelium and is a vasodilator. Last little tidbit. Previously, we discussed that arginine is also involved in the synthesis of creatine in products derived from the amino acid glycine and urea in the urea cycle. Arginase enzyme catalyzes the formation of urea and ornithine, an amino acid regenerated and required for urea biosynthesis. During the reaction catalyzed by inducible form of nitric oxide synthase, which produces nitric oxide, another amino acid, citrulline, is formed, which is also an intermediate in the urea cycle. This is just kind of tying it all together, talking about arginine, how within this formation, the formation of the nitric oxide, we also that's arginase is the same enzyme, catalyzes the formation of urea and ornithine, which is part of that whole process that we looked at within the urea cycle. Just kind of showing the link. Okay, that is it for chapter 9. Thank you for staying all the way through. If you have any questions, leave them in the comments below. I'll try to get back to them as soon as I can. And yeah, good luck studying. Hopefully, maybe if you want to watch this back as well, just taking some of the notes of what specific things that I mentioned to review, to study, and go ahead and hopefully you, at this point, you've made that little cheat sheet and highlighted those portions that I told you to focus on for your exam. Thank you for watching and have a great day.